Today, we're focusing on cellular respiration, which is the process that allows living cells to extract energy from food. This is one of the central pathways in biology because it connects what organisms eat to how their cells function. The energy stored in glucose and other nutrients does not power cells directly. Instead, it must be converted into ATP. As we move forward, we will trace how that conversion happens and how this process links together metabolism, oxygen use, and energy production inside the cell. All life requires a constant input of energy, and in most ecosystems, that energy can be traced back to the sun. Sunlight is captured by plants during photosynthesis and converted into chemical energy stored in sugars and other organic molecules. That stored energy does not stay locked inside plants. It moves through ecosystems as organisms eat plants or eat other organisms that have actually eaten plants. At the same time, matter is constantly recycled. Atoms like carbon, oxygen, and hydrogen move from the atmosphere into the organisms and then back into the environment. Energy flows in one direction through ecosystems, but chemicals cycle over and over again. Cellular respiration is a key part of the cycle because it releases energy from food while returning important molecules back to the environment. Plants and other autotrophs, which literally means self feeders, have the remarkable ability to build all of their organic molecules from completely inorganic ingredients. They take in carbon dioxide from the air, water, and soil, and minerals such as nitrogen and phosphorus. Using energy from the sunlight, they rearrange those simple molecules into complex organic molecules like glucose. That glucose becomes the foundation for everything else in the plants, from cellulose in the cell walls to stored starch. Heterotrophs, on the other hand, cannot build organic molecules from inorganic sources. Humans, animals, fungi, and other microorganisms must consume other organisms to attain organic compounds. We rely on the work of autotrophs either directly by eating plants or indirectly by eating animals that have eaten plants. This division between autotrophs and heterotrophs sets up the entire energy structure of ecosystems and makes cellular respiration essential for all organisms. Most ecosystems on Earth depend almost entirely on photosynthesis as the foundation of their food supply. Without plants and other photosynthetic organisms capturing energy from the sunlight, there would be no steady input of chemical energy into the system. Biologists call these photosynthetic organisms producers because they produce the organic molecules that fuel life. In contrast, heterotrophs are called consumers. They cannot make their own food from inorganic materials, so they obtain energy by consuming plants or by eating animals that have eaten plants. Even top predators ultimately depend on producers at the base of their food webs. Photosynthesis begins with two simple molecules, carbon dioxide and water. Carbon dioxide enters the plant from the air through tiny openings in the leaves called stomata. These pores allows gases to move in and out, connecting the plant to the atmosphere. Water is absorbed in the soil by the roots and transported upwards through the specialized vascular tissues in the leaves. Inside the leaves, chloroplasts capture light energy and use it to rearrange the atoms of carbon dioxide and water. Through a series of chemical reactions, these simple molecules are converted into glucose, a six carbon sugar that stores chemical energy. Along the way, oxygen gas is produced as a byproduct and released into the atmosphere. This oxygen is the same oxygen that organisms use during cellular respiration, completing the biological connection between these two processes. Cellular respiration uses oxygen to break down sugars like glucose and convert that stored chemical energy into ATP, which is the energy cells can use. In both plants and animals, most of this ATP production happens inside the mitochondria. As glucose is broken apart, carbon dioxide and water is released as waste products. These same molecules are the raw materials for photosynthesis, which shows us how tightly connected these two processes are. Plants also often make more glucose than what they need right away. The extra energy can be stored as starch, like in potatoes, and used later for growth, development, and survival. Cellular respiration is an aerobic harvesting of chemical energy from organic molecules like glucose. When we say aerobic, we mean this process requires oxygen. It is the primary way cells convert this energy stored in food into ATP, the usable form of energy that powers cellular work. Because it depends on oxygen, cellular respiration requires a constant exchange of gases from the environment. Cells take in oxygen, which is essential for the final step of energy production. At the same time, they release carbon dioxide as a waste product. That carbon dioxide must be moved from this organism. This continuous exchange of oxygen in and carbon dioxide out is what keeps the entire process running efficiently. Cellular respiration is not a single reaction, but a series of many coordinated chemical reactions. To make it easier to understand, we group these reactions into three main stages: glycolysis, the citric acid cycle, and the electron transport. Each stage plays a specific role in the gradually extracting energy from glucose. Instead of releasing all of the energy at once, the cell captures it step by step. This controlled breakdown allows energy to be transferred efficiently into ATP. The first stage, glycolysis, occurs in the cytoplasm. During glycolysis, one 6 carbon glucose molecule is split into two 3 carbon molecules called pyruvic acid, or pyruvate. After this, the citric acid cycle, also known as the Krebs cycle, completes the breakdown of the fuel. This stage takes place in the mitochondrial matrix. As glucose is fully broken down, carbon dioxide is released as a waste. By the end of the citric acid cycle, most of the carbon atoms from glucose have been removed and released as carbon dioxide. Glycolysis and the citric acid cycle produces a small amount of ATP directly, but the most important role is transferring high energy electrons to a carrier molecule known as NAD+. When NAD+ accepts electrons, it becomes NADH. NADH acts like a shuttle, carrying those high energy electrons to the next stage. The electron transport chain, this is where most ATP is produced. The cell is essentially collecting energy in the form of electron carriers and delivering to the final stage for maximum ATP generation. The third stage of cellular respiration is electron transport. In this stage, the high energy electrons carried by NADH are passed through a series of proteins embedded in the inner mitochondrial membrane. As these electrons move step by step through the chain, they gradually lose energy. That released energy is not wasted. Instead, it is used as power that production of ATP. At the end of the chain, oxygen acts as the final electron acceptor. Oxygen combines with electrons and hydrogen ions to form water. Without oxygen, this final stage cannot occur. The overall equation for cellular respiration shows that glucose and oxygen are rearranged to form carbon dioxide and water. The atoms are simply reorganized, but during that rearrangement, energy is released and captured in the form of ATP. The primary purpose of cellular respiration is to generate ATP for cellular work. On average, each molecule of glucose produces about 30 to 32 ATP molecules depending on the cell and its condition. Glycolysis is the first stage of cellular respiration and occurs in the cytoplasm. One 6 carbon glucose molecule is split into two 3 carbon pyruvate molecules. This process requires an initial investment of 2 ATP molecules, but it produces 4 ATP in total, resulting in a net gain of 2 ATP per glucose. During glycolysis, high energy electrons are also transferred to NAD+, forming NADH. Even though glycolysis produces only a small amount of ATP, it is essential because it begins the breakdown of glucose and prepares molecules for the next stage. Before pyruvic acid can enter the citric acid cycle, it must be broken down and converted into a more usable form. Each pyruvate molecule loses one carbon as carbon dioxide. The remaining 2 carbon fragment is called acetic acid. During this conversion, high energy electrons are transferred to NAD+, forming NADH. The 2 carbon fragments then attaches to a molecule called coenzyme A, forming acetyl coenzyme A. This step is crucial and critical because acetyl coenzyme A is the molecule that actually enters the citric acid cycle and allows this process to continue inside the mitochondrial matrix. The citric acid cycle completes the breakdown of the fuel. Acetyl coenzyme A combines with a 4 carbon molecule to form a 6 carbon compound called citric acid. As this cycle proceeds, carbon atoms are removed and released as carbon dioxide. For each acetyl coenzyme A that enters, two molecules of carbon dioxide are released as waste. During these reactions, energy is harvested and transferred to electron carriers. A small amount of ATP is produced directly, but most of that captured energy is stored in NADH and FADH2. Although the citric acid cycle produces only a small amount of ATP directly, its main function is to load up the electron carriers NADH and FADH2 with high energy electrons. These carriers will deliver their electrons to the electron transport chain in the next stage. All of the carbon atoms that entered as fuel are eventually released as carbon dioxide. The original four carbon acceptor molecule is regenerated, which allows the cycle to continue turning as long as the acetyl coenzyme A is available. During cellular respiration, electrons removed from the glucose and other food molecules gradually fall to a lower energy state. First, those electrons are transferred to NAD plus, forming NADH. NADH then donates those electrons to the electron transport chain, which is the series of carrier proteins embedded in the inner mitochondrial membrane. As electrons move through this chain, they release small amounts of energy at each step. This stepwise release allows the cell to capture energy efficiently instead of losing it all at once as heat. The overall movement of electrons during respiration can be thought of as a downhill journey. Electrons start in glucose, are then transferred to NADH, then move through the electron transport chain, and finally end up in oxygen. Oxygen acts as the final electron acceptor. This downward flow is essential because it drives the energy production process. Without oxygen at the end of the electron transport chain to accept electrons, the entire system would back up and ATP production would stop. The electron transport chain is located in the inner membranes of the mitochondria. This membrane is highly folded into structures called cristae, which greatly increases the surface area. Most surface area means more electron transport chains can be embedded in the membrane. This structural design allows the cell to maximize ATP production. The energy released as electrons move through the chain is used to create a buildup of hydrogen ions, setting the stage for the final step of ATP production. Inside the inner mitochondrial membrane are remarkable protein structures that function like tiny turbines. These structures are called ATP synthase. They sit next to the proteins on the electron transport chain. The energy that was originally stored as NADH and FADH2 is now used to power these molecules' machines. As energy is released during electron transfer, it drives the production of ATP. ATP synthase is the enzyme responsible for actually attaching a phosphate group to ADP, forming ATP. This is where most of the ATP from cellular respiration is generated. Cellular respiration can generate up to 32 ATP molecules per glucose molecule under ideal conditions. This makes aerobic respiration highly efficient compared to anaerobic pathways. The majority of ATP is produced during electron transport and chemiosmosis. What begins as chemical energy stored in glucose has now been converted into ATP, which fuels processes like muscle contractions, active transport, and biosynthesis. Cellular respiration is the central pathway that allows cells to transform food into usable energy. Cellular respiration is not limited to burning glucose. It is more like a versatile metabolic furnace. Carbohydrates, fats, and proteins can all enter the pathway at different points. For example, fats can be broken down into molecules that feed directly into the citric acid cycle. Proteins can be converted into intermediates as well. This flexibility allows the body to maintain a balance of metabolism depending on what nutrients are available. All of these pathways interact to keep ATP production steady. Some cells can function temporarily without oxygen. This process is called fermentation, which is an anaerobic pathway. Fermentation relies only on glycolysis, which does not require oxygen. Glycolysis produces two ATP molecules per glucose, but without oxygen, the electron transport chain cannot operate. Fermentation allows the cell to regenerate NAD+, so glycolysis can continue. Although fermentation is far less efficient than aerobic respiration, it provides a short term solution when oxygen is limited. For glycolysis to continue, NAD plus must be available to accept electrons. Under aerobic conditions, NAD plus delivers its electrons to the electron transport chain, and NAD plus is regenerated. However, under anaerobic conditions, oxygen is not available, so the electron transport chain cannot function. In human muscle cells, NADH transfers its electrons to pyruvic acid instead. This produces lactic acid and regenerates NAD+, allowing glycolysis to continue producing a small amount of ATP. This process is critical during short bursts of intense activity when oxygen delivery cannot keep up with demand. Yeast and some other microorganisms can survive with or without oxygen. Under anaerobic conditions, they perform a fermentation to keep glycolysis running. Instead of producing lactic acid, many microorganisms produce ethyl alcohol, also called ethanol, along with carbon dioxide. That the specific waste product of fermentation varies by species. This metabolic flexibility allows certain organisms to thrive in environments where oxygen levels fluctuate. Both aerobic and anaerobic respiration begins with glycolysis. That makes glycolysis the universal energy harvesting pathway of life. No matter the organism, no matter the oxygen condition, glycolysis is always the starting point. There is an evolutionary reason for this. Between 3.5 to 2.5 billion years ago, Earth's atmosphere contained little to no oxygen. Early prokaryotes could not rely on oxygen dependent pathways because oxygen was not available. They likely generated ATP exclusively through glycolysis. The fact that glycolysis is found in almost all organisms today strongly suggests it evolved very early in the history of life and became the foundational metabolic pathway that everything else was built upon. Glycolysis occurs in almost all organisms from bacteria to humans. That widespread presence suggests it evolved very early in ancestors common to all domains of life. When we see a pathway shared across such diverse organisms, it tells us that it must have been established before those lineages split apart. Its location in the cell also supports this idea. Glycolysis takes place in the cytoplasm and does not require membrane bound organelles like mitochondria. These organelles evolved much later, more than a billion years ago, but the first prokaryotic cells. The simplicity and universality of glycolysis points to an ancient origin and its foundational role in cellular metabolism.